experiments. Without experiments in science, they don't agree. Without proof, how can you agree something, right? So that was the problem. So the solution for this problem, that is, how does DNA replicate? So the semi-conservative. So for that, a proof was given by Measles and Stehel. Okay, so we say that Measles and Stehel's experiment, very famous experiment. Okay, it is a very smart experiment again. So what they do is, we know that DNA is made up of nitrogenous bases. Correct, more people. So DNA's composition is what? Nitrogenous bases. So nitrogenous bases are what? Your nucleotides. You have your adenine, thymine, cytosine, and guanine. So they have their nitrogen in their structure. So what we can do is, what they did was, they decided that we can grow the bacteria. Okay, which bacteria they choose? They choose E. coli. So they chose E. coli and they thought that, okay, I can grow E. coli using two different types of, lighter isotope and heavy isotope of nitrogen containing media. So what is the meaning? So we know that the normal nitrogen that we see in our environment is N14, correct? No? Whereas they chose another media which contains a heavier isotope, that is N15 nitrogen. Okay. So N15 nitrogen, how do they incorporate? Means, why do they need this? See, when the DNA is made up of nitrogenous bases, correct? No. So these organisms definitely need this element called nitrogen to construct some material. So we can provide this nitrogen through what? Ammonium chloride. So NH3Cl it is, okay, not NHCl. NH3Cl was used. So in NH3Cl, you can see that the nitrogen is of heavy isotope. And other NH3Cl, what they used is of lighter isotope, that is N14 and N15. So now what they do is, they grow E. coli on N15 media for several generations, and normal N14 media also for several generations. Okay. Now N14 they understand. By understanding, we go to the centrifugation, we understand that these cells are quite heavier. So they keep it aside. Now it comes to N15. Now once they are sure that 100% of the cells are carrying N15 nitrogen in their DNA, what they do is, they shift this N15 E. coli into N14 media. Okay, see what we do? First, we grow the E. coli in N15 media, correct? No? And after several generations, you have the proof that it is 100% N15. We transfer this N15 cells into N14 containing media. That is our normal media. So once we transfer these cells from N15 media to N14 media, now what happens? You see, E. coli cells, they divide in every 40 minutes. For every 40 minutes, you can see one E. coli becoming two. Okay. But in between, every 20 minutes, okay, in every 20 minutes, what is happening? One replication cycle will finish. That means for every 20 minutes, a parental strand will produce two daughter strands, okay, two new DNA. So that is happening. So now what they did, the moment they transferred this N14 E. coli N15 E. coli into N14 E. coli, okay, N14 media, what happens? They measure the time. You can see that the time frame is divided into 0, 40, 60, 80. So these numbers represent time in minutes. Okay, these are minutes. So what are we doing? First, the moment we add this N15 into N14, we subject it into density gradient centrifugation. So what is density gradient centrifugation? You know that the centrifugation is a technique that we use to separate molecules or particles which is suspended in a liquid, okay, based on their mass by volume. Okay. Now each of this molecule, we have a, what do you call it, a density, a particular density is there. Now you can ask me, why don't we use water? Why don't we use other what do you tell, other compounds as a gradient, the gradient for centrifugation? Why only cesium chloride? See, when we use cesium chloride and we subject it for centrifugation at 50,000 RPM, okay, 50,000 RPM means you spin at 50,000 revolutions per minute, like this, okay, for around 7 to 10 minutes. When you spin like that, what will happen? It ensures that the DNA separates from the cell. You get my point? So the cesium chloride helps the DNA separation based on the density. So what we do, we take the cells, the moment we add from N15 to N14 media, and we subject it to density gradient centrifugation. What do we use? We use cesium chloride. Remember this. So the cesium chloride is a gradient that we use for separation. Understood? No. Now, once we have this cesium chloride separation, see, at the 0th minute, the moment I add, what I did the centrifugation. So in my centrifugal tube, what I can see that there is a band. this band is 100%. You can see 100%, there is only one band and it is at the certain depth. Okay, it is at the bottom, almost at the bottom. Now what I'll wait for another 20 minutes, exact 20 minutes. I'll take the cells, okay, and then what I will do is I will again subject it for centrifugation. Now this time something unique happens. You can see here at the 0th minute when I did the centrifugation, you can see that there is a 100% one band. Correct, no people? There is 100% one band here. This is what? This is N15 band. Got it? No, N15, N15. You can see that we have two DNA strands, okay, in each DNA. So both this DNA is grown in N15 media. That is why both the strands will have N15, N15. That is nitrogen in this each strand is of heavy isotope. Now after 20 minutes, what happens? One strand of N15, which is the parent strand, is used and a new strand is constructed along with it, okay, using this as a template. Now we get a N14, correct? No, N14. Sorry, this is not quite pretty, it's ugly. Anyways, sorry. Here it is, okay? N15 is a parent strand. Now what will happen? A new strand that is synthesized will be N14. You get my point? Now you can see here, this strand is old and the light yellow is the new one. Here it is old, this is the new one. Now what do we have, people? When I do the density, again, if I do the density gradient centrifugation, I will get a band slightly above it, but only one band. What kind of band I will get? I'll get an intermediate band. Why an intermediate band? You can see that here, both the strands, okay, are having N15, N14. One heavy, one light. Again, one heavy, one light. So since it contains one heavy, one light, one heavy, one light, now what will happen? It will just float a little bit above this N15, N15. You get my point? Now what I will do? I'll wait for another 20 minutes. After 20 minutes, it is at the 40th minute, I will go for another density gradient centrifugation. Now what happens? Now I will get two separate bands. Now what happened in the 40th minute? You can see that this old strand and the new strand gets synthesized. This is the old strand and this is the new strand. This is the old strand. This is the new strand. Again, this is the parental strand. This is the new daughter strand. Correct? No. Now what happens? These two are belonging to N15 and 14. And here, 1, 2. Again, these two are belong to what? N14, N14. Both of them are quite light. That is why I get 50%. Okay, 50% of the strands are of N15, N14. And 50% of the strands are of N14, N14. Clear? Now I go for another cycle at the 60th minute. When I go at the 60th minute, now what happens? You can see here. Parent strand, daughter strand. Parent strand, daughter strand. Parent strand, daughter strand. Parent strand, daughter strand. Again, similarly, you can see here there is a proper, like this, right? So like this, it keeps on going. Now you can see that only 2 of the strands out of 8, 1, 2, 3, 4, 5, 6, yeah, out of 8 belong to what? N14, N15. That is why, see, 2 out of 8 is what? 25%. So 25% is N15, N14. And then 75% is what? It is N14, N14. So it is lighter, no, it will come up. So now I will go for another cycle, the last cycle at the 80th minute. So if I go at the 80th minute, now what will happen? Out of 12 strands, 1, 2, 3, 4, 5, 6, I don't know. Okay, so when you just do this, you can see that only 2 out of the whole bunch is of N14, N15, whereas the rest is of N14, N14. So now you can see that as the time passed, okay, first it was 100%, then a new band. It is exactly becoming half and half and half. So that is why the percentage decreases from 100, 50, 50 to, increases, let's say. 50 to 75, 75 to 88. Similarly, in the opposite side, you can see that there's a gradual decrease. It goes from 100 to 50, 50 to 25, 25 to 12.25, something like that. So mathematically seeing, only if the DNA segregates through semi-conservative model, you can get these numbers, correct? No, you can get that this proper separation or division of half. So that is why this experiment gave a solid proof that the DNA replicates through semi-conservative mode of replication. Okay, so you can try disruptive. You can use any numbers, but you won't end up with the every cycle, like even though you conduct the experiment 100 times, you always get the same numbers. But if you see disruptive, okay, if you see other mode of conservation, okay, other mode of replication, you don't find a similar distribution of the chain of the what do you tell numbers. So like 50 to 75, 75 to 88. This doesn't come in any of these two models. So that is why semi-conservative mode of replication, gained a solid proof through this experimentation. So did you understand, guys? Yeah. Are you following me? Are you able to listen? Are you able to understand? Is it fast? Yes, no? So, as you guys type in the message, I will just move on. Okay. Now comes the mechanism of DNA replication. This is quite fun, and let's see what is happening here. Okay. Now, we understand that what is DNA replication. In DNA replication, it includes the process of, one we have a parental DNA. It makes the copies. One DNA becomes two. That is, it creates two daughter DNAs. So, chalo, we'll see how the process happens. If you look at the whole DNA, as I said, the whole DNA is quite not accessible if you look at the whole structure. Now, in you prokaryotes, if you see, the DNA is quite circular. That is, even the chromosomes are quite circular. Okay, they are circular arrangement you can find. And if you see the points at which we can access the DNA to use it for replication, you can't use any points. That means the cells cannot use any place in the DNA to start the process of replication. So to start the process of replication, there are something called as conserved sequences. Okay, conserved sequences. So this whole mechanism of a replication is very complexly wired within the cell. So let's not go how the cell initiates the process of replication. Okay, through a lot of signaling. It's a very, very, very deep topic. Let's not go there. But let's consider that the cell has decided that it is time for me to replicate my own DNA. Okay, I have started. I've decided. Now what happens? I told you, once the cell decides that it's time for the replication cannot start at any point in the DNA. Okay, so it has some sequences, and this I call it as a conserved sequences. Now, what are conserved sequences? These conserved sequences are not for today. It is coming from generation to generations for millions of years. That's why we call it as conserved. That means it's been existing throughout the time. Okay. So now these conserved sequences, okay, what is the use of it? So these conserved sequences are also called as ORI sites or origin of replication. ORI site is also called as origin of replication. So these sites are responsible for accepting the proteins that are responsible, that are included or that involves in the process of DNA replication. You understand my point? So these sites, what they do, they allow this place for the assembly of this machinery, okay, protein machinery, to come and start the process of replication. So that's why ORI site becomes a very significant part in which the DNA replication can be initiated. If you look at prokaryotes have only one ORI, that is only one origin of replication, whereas eukaryotes have many origin of replications, correct? So prokaryotes have only one origin of replication, whereas eukaryotes have many origin of replication. So that is one small thing that we have to understand. Now, coming to the mechanism of DNA replication is very complex and it involves several assembly of proteins. So here is a list of proteins that are involved in both prokaryotes and eukaryotes to generalize. So what is the role? Okay, let's see. Now, if you see a general DNA replication in prokaryotes, it is a list of the proteins that is involved in this process. First, we will see the whole DNA process, okay, involves synthesis of new DNA. Synthesis means production of DNA, correct? No? For us to produce DNA, we require an enzyme called as DNA polymerase. Okay, DNA polymerase. So this DNA polymerase is a very effective enzyme that synthesizes both the strands. But if you look at the classification of DNA polymerase, like if you look at the DNA polymerase as an enzyme, what you can see is, you can see that DNA polymerase is not just of one type, it is made up of several different polymerases like DNA pol 1, DNA pol 2, okay, pol 2, DNA pol 3. Okay, these are the three different types of DNA subtypes of different DNA polymerase you can see in prokaryotes. But what about eukaryotes? Eukaryotes also use DNA polymerase, but we denote them as alpha, beta, gamma, delta, and epsilon. Sorry, my symbols are very bad, but you understand, right? In eukaryotes, we can see that the DNA polymerase is a little more complex, and it is made up of subproteins like alpha DNA polymerase, beta DNA polymerase, gamma DNA polymerase, delta DNA polymerase, epsilon DNA polymerase. So 5 types of polymerase are there in eukaryotes. But generally, when we study DNA, DNA replication, we come across this very familiar DNA polymerase in prokaryotes. That is, DNA polymerase has DNA pol 1, pol 2, pol 3. Now let us look at the roles of this. You understand? So the DNA polymerase has a very specific role. Now what is that? We have DNA polymerase 1, DNA polymerase 3 is here. So since you get a little bit confused, let me go back to the process so you will understand very well. Let's go with DNA pol 3 first. Okay, our very important enzyme is what? Everything is important, but we can see that this is involved in a larger process. Okay, now let's look at DNA polymerase. What is the role of DNA polymerase 3? Now, DNA polymerase 3 or any DNA polymerase, if you see, it cannot, okay, it cannot start replication. Okay, so what am I saying? I'm saying that the DNA polymerase, if I have a DNA, okay, let's say that I have a strand of DNA, correct? No, this is the parental DNA. And now I have to start the replication. That means this is A, I have to start adding T, C, G, whatever is that. This. So you can see that the DNA synthesis has to start from the first base pair, correct? No? So this does not happen, okay, with the use of DNA polymerase. The DNA polymerase cannot start the synthesis of DNA from the first base pair. It's not possible. So this synthesis is called as de novo. Okay, de novo. What is de novo? I cannot do it on my own. De novo is doing it on your own, but DNA polymerase cannot do it on their own. So if they cannot do it on their own, how do they do it? You can see that the DNA polymerase requires a free 3' OH. What is the meaning of this, guys? We know that each of our nucleotide is having a pentose sugar, and the pentose sugar has a third carbon atom. At the third carbon atom, there is a OH group. At the fifth carbon atom, there is a phosphate group. Correct? No? We already are aware of it. So our DNA polymerase required this free 3' OH to start the replication. You understand? So where do they get this free 3' OH? They get this with the help of something called as with the help of something called as a primer. Okay, a primer. We'll discuss what is a primer. So let us imagine that a primer is helping our DNA polymerase to provide a 3' OH. Now it comes, what is the role of DNA polymerase 3? Now our DNA polymerase 3 has two important functions. What is that important function? Now the important function is, first one, it synthesizes from 5' to 3' a new DNA, okay, new DNA continuously, okay. So it synthesizes a new DNA in 5' to 3' direction, okay. That is the first function of DNA polymerase 3. What is the second function? The second function of DNA polymerase is, see, at one point you can see that there is a DNA and there is a DNA, there is a DNA, there is a DNA, okay. So you can see that there are two strands here. One we call it as a leading strand, other one is called as a lagging strand. So the one strand where the continuous synthesis of DNA happens, that is called as 5' to 3' strand or a leading strand. Here what happens, DNA polymerase 3's role is to only synthesize new DNA. That's it. But in the opposite strand, what is happening? It has a different role. The different role is there is a DNA here, there is a DNA here, and there is a DNA here. What you can see is there are fragments of DNA. So these fragments of DNAs, these gaps have to be filled. So this filling of gap is called as DNA primer extension. I know that this is little complex, but you try to remember the role. When we go for the mechanism, you will understand what I'm trying to talk about. One function, it's see, you have here, no? You can see that this is the leading strand. 5' to 3', what is happening? You can see that the new DNA is synthesized very continuously. But if you look at the lagging strand, what is happening here? You can see that there are fragments here. Fragments of DNA will be there. So these fragments of DNA extension, like this new DNA will be filled in the gaps. So that extension is called as DNA primer extension. So 2 roles of DNA polymerase 3 is done. Let's go for another DNA. What is happening? What is the role of another DNA polymerase? The other important DNA polymerase we are talking about is DNA Pol 1. DNA Pol 1. So what is the role of DNA Pol 1? Now I told you we have RNA. There is a primer. So let us say that we have RNA, RNA, RNA. So the role of DNA polymerase 1 is to put a new DNA RNA primer. Okay, they make what? A new DNA RNA primer. So now what's happening, guys? We have DNA Pol 1. The DNA Pol 1, what it does? It removes RNA primer and it replaces with a new DNA. What is the DNA polymerase 3 doing? It is synthesizing the nucleotides continuously in 5' to 3' direction in leading strand. In the lagging strand, what it is doing? It is filling the gaps, wherever the DNA is missing. So these two are done, very simple. Now we have DNA Pol 2. DNA Pol 2, what it is doing? It is doing a very important function called as repair or cross-checking. It will do the cross-checking so that there are no mistakes. So let's fast forward a little bit because I know that it is quite hard for you to get it in the very first go. So I will go to the mechanism slowly as fast as possible so that we will understand very clearly. Clear, no? Next one is helicase. The role of helicase is it is the first one to come and recognize the ORI site. It will come and see that this is the place I have to start the replication. It comes at the ORI site. Once you see the ORI site, what you see? The DNA is made up of 2 strands. Correct? Yeah. The DNA is made up of 2 strands. So these 2 strands are both parent strands. So they are locked with each other. So this locking is done by what bonds? Hydrogen bonds. So now helicase comes and sits here. Now what it will do? It will hydrolyze. Okay. So once it hydrolyzes, so these bonds get added with water. So when the bonds are added with water, they start opening up. So helicase, what it will do unzipping. So it will unzip the 2 strands and it will open. Okay. Next one is ligase. Ligase is also called as a gum. Okay. Or it will fill the gaps. It will stick one DNA with other DNA. Okay. Next one is a primase. Primase is something that is a small startup or a heads up, or you can also call it as a push that it gives to start the DNA replication. Okay. Next, we have sliding cam, topoisomerase, and SSBPs. Okay. So instead of going into the roles, I clearly think that we have to go to the process. So it becomes easy, very easy for you to understand. Okay. So let's go here. Erase. Oh, 1 second, guys. Yeah. So now let's see what is happening. Let's imagine that we have a DNA and these both DNA are parental DNA. I'll write it as P and P. One strand is going from 5' to 3' direction. Other strand is going from 3' to 5' direction. This is the direction of 2 strands, correct? No? Now, these both of them are attached to each other or they're linked to each other, through bonds. Both the strands are attached. Now, the first enzyme that comes and assembles at the ORI site is helicase. Okay. Helicase, what it will do? It will recognize where I should start the replication and it will come and bind to the ORI site. Once it binds to the ORI site, the role of helicase is to open up the strand. Now, you can see here, excuse me. Now you can see here, this is the DNA strand. Once the helicase has come in the place, both the strands open up like this. Once they open up like this or unzip like this, right? Unzip like this, the strands are open here. That means the bonds are clearly not interacting with each other, okay? And they're just removed or separated apart. So once they're separated apart, you can see helicase is moving in this direction. So once this separation happens, now what happens, guys? Now, the crucial thing happens. When the bonds or when the nitrogen bases are separated, they becomes very unstable. So, and the moment the helicase moves forward, these bonds, they will get back to each other and they will again stick it. Okay, again the 2 parent strands will come and join. So that should not happen. So again, it should not become double strand once it unzips. So to make it stable, a special assembly of proteins comes here. What is a special assembly of proteins called as? It is called as SSBP. What is SSBP? Single strand binding protein. Okay. Now you can see here. What is SSBP? SSBP is called as a single strand binding proteins. Okay. Now the role of SSBP is to make sure the a rebinding of the DNA should not happen. So now what will happen? All these SSBPs will come and sit here and they will stabilize both the parental strands which are separated. So once this is done, as I told you guys, we do not have our DNA polymerase do not have the capacity to synthesize the DNA on its own. So what it will do, it will take the help of something called as a primase or a primer. So what is a primer? What is a primer? A primer is something that initiates the DNA replication. Okay. Now, what kind of primer we have? We have an RNA primer. What is an RNA primer? RNA primer is a short sequence of RNA, okay, which is synthesized by an enzyme called as RNA primase. So what kind of RNA primase? RNA primase or DNA polymerase means it has the capacity to synthesize RNA. Okay, so this RNA primase is a very unique type of RNA polymerase. Okay, RNA polymerase. So what kind of polymerase it is? It is a DNA dependent. It is a DNA dependent RNA polymerase. Okay, the nature of this enzyme is it is a primase. What kind of a primase? It is an RNA primase. What is a RNA primase? It is a primase that helps in synthesizing a short sequence of RNA, which we call it as RNA primer. So what kind of an enzyme it is? It is a DNA dependent RNA polymerase. Okay. Now what is the role of this enzyme? The role of this RNA primase is, let's say that we have two strands, the parental strands, correct? No, this is also parental strands. This is also parental strand. Let's say that this parental strand is going from 3' to 5' direction, and this strand is going from 5' to 3' direction. You see, now if our helicase is moving in this direction, now you can see that there is a 3' and 5'. Now what happens, guys? Always the DNA synthesis happens in what direction? 5' to 3'. Always. Always the direction of replication is what? From 5' it has to start and it has to go towards the 3'. That means the new DNA strands will be start synthesizing here, 5' to 3'. Now I told you we need a help of a RNA primer. Now what is the role of RNA primer? This RNA primer comes here. Let's say that we have base pairs here. Okay. Let's say that this is A, T, C, G, G, C, C. Okay. Now what is our RNA primer doing? RNA primer comes and adds a short RNA sequence. A. In RNA, you don't have A, T, C, G is in DNA. Okay, this is DNA. And if you see RNA, it is AUCG, guys. So all are same except for thymine. Uracil is replaced in RNA. Okay. Now A is there. A's complementation is what? T. But T will not come here. Why? Because it's an RNA sequence. It will be U. It will be A. It will be G. It will be C. C. G. G. Like this, a short RNA sequence is synthesized. How big is the sequence? Around only 10 base pair. Around 10 base pair sequence is synthesized first. So who does this? RNA prime. Now, why do we do this? If I synthesize, even though if it is 10 base pairs, the last base pair, this is 1, this is 10. Let's say this is 5', this is 3'. So at the 3' and what do I get? I get a free OH group. So when I get a free OH group, what does it mean? It means that my DNA polymerase is very, very happy. Why it is very happy? It can synthesize the DNA continuously now. Okay, it can start synthesizing the DNA continuously now. Are you following me, guys? What's happening? Are you understanding? You are very quiet today. No questions till now? Okay, chalo. Did you understand? RNA primer came. Okay, let me draw here. Now this is the place where our DNA strand has opened up. Got it? This is the 3'. This is the 5' parent strand. Now this is the 5'. This is the 3' daughter strand. Now who came here? An RNA primase came and synthesized the short RNA primer. In what direction? 5' to 3' direction. Now what do we have here? A free OH group. Got it? Now what happens, guys? Now DNA polymerase is very happy. DNA poly is very, very happy. Now guys, listen. For any synthesis, what do we need? Monomers. Correct? No, we need enzyme and we need a monomer. Because it is a polynucleotide chain. No, it is a polymer. Similarly, even RNA is a polymer. Both of them are made up of nucleotides. So we need monomers. What kind of monomers we need? See, DNA polymerase role is to synthesize new DNA, no? So they get this DNA through what? They need ATP, GTP, CTP, and TTP. What is this? Adenosine triphosphate, gynosine triphosphate, cytosine triphosphate, thymidine triphosphate. So these are the monomers that is coming for the assembly. So what happens to this? So this will become adenine, thymine, cytosine, guanine, which gets into the assembly of constructing a new DNA. These are the monomers. You understand? So this is what our DNA polymerase require. So DNA polymerase is an enzyme, and DNA polymerase require the raw material. So what is the raw material? This is called as DNTPs. What is DNTPs? Deoxyribonucleotide triphosphate. Okay, no? Deoxyribonucleotide. That is the name of the DNA. And triphosphate, because you see triphosphate, three phosphate groups. Correct? No? Why it is in the triphosphate form? I'll explain it. You get my point? So now DNTPs are required and we need the DNA polymerase. Okay, to start the process further. Now you can ask me a question. So what happened in RNA primer? What is RNA primer made up of? RNA primer is made up of RNA, polynucleotides. What are RNA polynucleotides? You have ATP, GTP, CTP, and UTP. Uracil triphosphate. You see, you can clearly see the difference. TTP and UTP. So they help in putting the RNA. You understood? So now what happened, guys? We have the DNTPs and then we have the DNA polymerase 3. Okay. Now we have the DNA polymerase, and this DNA polymerase we are using is DNA polymerase 3. I told you what is the role of DNA polymerase. It uses the free 3' OH, okay, which is in the leading strand, okay, and it synthesizes a new DNA strand continuously. A new DNA strand will be synthesized continuously in what direction? 5' to 3' direction. So this is one rule. Done. Clear? So now what is happening in the lagging strand? You can see here very clearly. Now I will write here, okay? Now you can see here, one is going from, you can see, this is a 5' to 3' direction, okay? Now this synthesis is happening in a continuous fashion, like we saw here. But what is happening in the opposite strand? The opposite strand, parental strand, is from 5' to 3' direction, whereas the daughter strand has to be synthesized in the opposite direction, 5' to 3'. Now there is a small challenge. What is this challenge? The challenge for our DNA polymerase is, let's say that this is the double bonds that are coming further. Now you see the DNA is opened up like this, and the helicase is moving in this direction. And as it moves in the direction, let's say that this is the lagging strand, 5' to 3'. Now, if my DNA helicase is moving like this, and my new synthesis of the DNA has to happen in 5' to 3' direction, you can see as the DNA gets unzipped, now our DNA polymerase will not get continuously 3' OH. Correct? No? The DNA has to be exposed. After a little bit of exposure, we should synthesize more and more of RNA primers. I know that it is a little bit confusing to imagine. Okay, let me draw this so you can understand. Hold on. Yeah, chalo. 1 second, guys. I need space. So let's say that this is our lagging strand. Okay. This is our lagging strand. Now, what's happening in our lagging strand? What is the direction of lagging strand? 5' to 3'. Okay, this is the parental lagging strand. Clear? Now, let's say that we have to synthesize DNA from which direction? 5' to 3'. Correct? No, in this direction, the DNA replication has to happen, 5' to 3' direction. Now you can see, you can see here, if my new opening DNA, if my DNA is opened in this direction, now what will happen? As the new strand is opened, you can clearly see that there is no OH group in this direction. Correct, right? So what should happen? Our DNA polymerase will use the lagging strand and it will start laying a short RNA primers. So, in the lagging strand, once it is exposed, our RNA primase, what it will do is, it will use the newly exposed parental strand and it will lay the RNA primer in like this 5', 3', 5 prime, 3 prime. Short RNA primers are synthesized. Okay. So what are these? These are all RNA. Who is synthesizing the RNA? RNA primase. Okay. So once this RNA primase is done, now what is the role? Our DNA polymerase will come and it will start. So this is RNA, this is RNA, and DNA pol 3, okay, DNA pol 3 will start expanding this, okay, from 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime, like this. The RNA, DNA, RNA, DNA. Got it now? RNA, DNA. Like this, the short fragments of RNA, DNA, you can see in the lagging strand. Now, can we have RNA in our whole DNA? It's not possible, correct? No, we should not have any RNA in our DNA, whole DNA. So now we have to remove the RNA. If you look at the whole things that is happening in our DNA synthesis, one strand, in the leading strand, what we have here, we have RNA here, and then continuous DNA is here. But in lagging strand, it is completely different. RNA, DNA, RNA, DNA, RNA, DNA. Like this, it's completely messed up. So for that, what will happen? We have DNA pol 1. Okay. What do our DNA polymerase 1 will do? Our DNA polymerase will come, it will cut the RNA, it will cut the RNA primer, throw it away, and put the new DNA. So this is called as DNA primer. Okay. Got it? Again, in the lagging strand, it will come, it will cut off all this RNA, remove them, and then replace it with what? DNA. So now what do we have? We have DNA, DNA, and DNA like this. You got my point? So what happened here? We had two strands. One strand is called as a leading strand. Other strand is called as a lagging strand. In the leading strand, initially a short RNA primer was synthesized. And later what happened? A continuous daughter strand was synthesized. In the lagging strand, what happened? Since the direction of replication is in the opposite direction, we required continuous, okay, or frequent RNA primers. So this RNA primers was done by RNA primase. After that, these primers were extended by who? These primers were extended by DNA pol 3. DNA pol 3 will come and it will start synthesizing DNA, RNA, DNA, RNA, DNA. Okay, like this. Now what will happen? We should not have RNA in our DNA. So DNA polymerase 1 will come. So the role of DNA polymerase is endonuclease activity. Endonuclease activity. So what is the meaning of endonuclease? Endonuclease means it will cut the any part of DNA, okay, or RNA from the whole strand. So it will come, it will cut off RNA, it will also cut off RNA, it will remove all the RNA. And next what it will do? It will replace that space where the RNA is not there with the DNA, you can see. Now what we have here, in the leading strand, you can clearly see, okay, you can clearly see that there is a problem. Here was the short DNA that was synthesized by DNA polymerase 1. And then after that, there is a continuous DNA. But if you see in the lagging strand, this is DNA, 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 like this. There are in the form of fragments. Correct? No? So here it is leading. And here it is lagging. Got it? Here it is leading, here it is lagging. In the lagging, you can see there is no continuity in the new DNA form. But here you can see that there is continuous, but only one gap is present between a short DNA fragment and a long DNA fragment. Now, can we have gaps in the DNA? Definitely no. For that, what will happen? We have ligase. So DNA ligase will come. DNA ligase, what it will do is, it will take the phosphate, it will take the OH of the two different DNA strands and it will stitch. Okay. So it is like a molecular gum. So it will start sealing the gaps. So these gaps, correct? No, these fragments are called as Okazaki fragments. And the sealing of the Okazaki fragments is done by DNA ligase. Is it clear, guys? Are you following me? Please respond. Is it getting complex? Or should I explain in a more simple form? Are you listening to me? Can I have your response? Where did my screen share go? Hold on. Just a second. Uh, 1 second, people. I just missed my screen share. Yeah. So did you guys understand till now? Yeah. So we clearly in DNA replication, there are only few important points. Okay. But the process is very nice. We have to take it a little slow, but unfortunately, I have to rush a little bit. That's a major problem. I don't know if you are able to see the screen, full screen. Yeah. Yeah. So I told you the roles of DNA pol 1, DNA pol 2, helicase, ligase, primase, correct? No, SSBPs. Now, what is happening here? We understand that we need energy for this whole process, correct? No? So, who gets the energy? Where does the DNA gets energy to do all these things? So, I told you that then the monomers, that is DNTPs, DNA DNTPs or DNTPs, sorry, that is deoxy ribonucleotide triphosphate, whichever the DNTPs that are coming for the assembly are in the what form? They are in the triphosphate form. Okay. So whenever you break the bond, you know that ATP, GTP, they carry a lot of energy which is stored in the bond. So when they come and assemble with the DNA polymerase, this TPs, triphosphates are converted into monophosphates. So it releases a huge amount of energy. So this huge amount of energy is used for the synthesis of new DNA, okay, for the attachment or whatever it is. Okay, that is another thing. Now, if you look at the process, the unwinding of the strand is very fast. If you almost around 2000 base pairs per second, so it is a very tremendously fast synthesis that is happening with respect to prokaryotes. In prokaryotes, the DNA replication is very fast, but in eukaryotes it is little less. It is the speed is little less. Okay, reason being the DNA packing. The DNA packing in eukaryotes is very tight, whereas in prokaryotes it's quite loose. Okay, so that's why we can see that in prokaryotes the DNA replication is very fast. So assuming that they replicate at 2000 base pairs per second, okay, now you see that if you have two threads which are intertwined like this, like like the DNA, imagine you take the two strands and you hold it and start ripping apart. That means separating apart. What will happen as you move further? Okay, as you move further, the whole strand gets intertwined, and then you can see that there is a possibility of nicking. Okay, we call it as nicking. What is nicking? Nicking means breaking. Okay, so the force or the speed at which the helicase keeps on unwinding. Now, as the at the down end of the DNA strand, you can see that there is a kind of a stress that develops. So this stress is called as torsional stress. Okay, so the torsional stress, what it will do, it will lead to nicking of DNA. That means breaking of the parental strands. Okay, in the further end. So for that, a topoisomerase will come. A topoisomerase in prokaryotes and a gyrase in eukaryotes. Excuse me, guys. Okay. So what it will do, it will start relieving this torsional stress. So this relief of the torsional stress, what it will do, it will make sure that the DNA will not break. So where is the topoisomerase? If helicase is this, which is moving in this direction, the topoisomerase is the one enzyme that is before helicase. You understand? So it goes before helicase, making sure that the further DNA is not stressed because of the unwinding. Okay. So what is topoisomerase? Topoisomerase is a protein that helps in preventing nicking of DNA or breaking of the parental DNA. Got it? Yes. So this is another thing. Very important. Topoisomerase or gyrase. Now, once we have the leading strand and the lagging strand, what are the different aspects we need to consider? The question is, let's say that the strand is synthesized in a leading direction. Okay, this is the leading strand. A daughter DNA is a leading strand. What is the direction of the parental strand? If I have to call my daughter DNA as a leading DNA or the leading strand DNA, my parent strand should be in 3' to 5' direction. You can see here. My leading strand, parent strand is what? 5' to, sorry, yeah, 3' to 5' direction. So see, this is 3' and this is the 5'. So this is my parent strand. Whereas the daughter strand, the newly synthesized strand is in what direction? 5' to 3' direction. So this is daughter. If daughter is 5' to 3', parent should be what? 3' to 5'. So keep an idea on this. So in lagging strand, what is happening? The parent strand is always in 5' to 3' direction. Whereas the newly synthesized strand, again, it will synthesize from what? Opposite, 3' to 5. You understand? Even though we call it as leading strand and lagging strand, what you can clearly observe is it doesn't make any difference. Okay. Both of them almost finish the replication at the same time. Only few hundred base pairs are difference. But considering the volume of DNA, it's a very pretty fast process. All right. So this is the thing. What I will do is I will try to make it little more simple by bringing a table so that you can understand more clearly. Okay, I need an empty slide, so I could not gain the empty slide, so to write the diagrams and everything. Tomorrow, in the first place, what we'll do, we'll try to revise the DNA replication and make sure it goes in a flow. Okay. Now we are a little bit aware of the terms. So when I tell tomorrow again, it will be like a story. Okay. So I'll meet you guys tomorrow. And sorry for this. Okay. And have a nice day. If you guys have anything or any doubts, you can ask now. Clear? Sorry. Guys, I've launched a poll. Please try to answer. All right, guys. Thank you very much. I'll see you in next class. Okay. Yeah. See you.